Hi, my name is Julia, and welcome to my summer as an RU student at the Chicago Botanic Garden. This is me walking in on Monday morning, and I saw this cute deer, so I just thought I'd show you guys. So this is me working on processing some leaf samples that were collected last summer. What I have here is samples from 42 different species from different oak woodlands in the Chicagoland area. And what I'm doing is I'm taking 50 milligrams of sample that's already in those little envelopes that you see on the table, and then I'm putting them to 25 milligrams, and then each of those 25 milligrams is gonna go into a tube. So this is me walking into the genetics lab, and this machine is called a fast prep, and it is gonna be what turns our little bits of leaf into the powder that we're eventually gonna send for carbon and nitrogen analysis. So I'm just turning it on here. And then next up is I load this little holder. I do have to run 24 samples at a time, so I'm putting in all 24. And then I'm going to put the little holder into the machine. And it's important that I do this properly so that the machine runs and I don't lose any sample. So you do have to screw this top on so that it clicks twice. Once that is done, I am going to close this lid right here. Very important. And then I'm gonna start the program, which is what I'm showing you here. So essentially what's gonna happen is these tubes are gonna spin very, very quickly for 30 seconds at a time. And there are beads inside that is gonna help break up our sample so that we end up with powder at the end. So this is me about to start it. As you can see, it is very fast. Now that it's done, you can already see that these tubes are a bit green. That would be the powder along the side. So then now on Tuesday, this is my college first student. His name is Edgar, and I'm mentoring him this summer. We are back to the scale and processing more of these samples. We're also working on some soil texture work, which is us walking across. And then tube A has our sand, and then B has silt, and then C has clay. We are washing out these samples so we can start them again. And these samples came from oak woodlands in the area, and we're looking at water holding capacity by seeing how much of different soil types are in each sample. So on Wednesday, I am by myself again, and this is me going into the ecology lab, and I am setting up to do more soil texture work. This took me pretty much all day. So when I wasn't doing soil texture work, I was working on my laptop doing some computer work. So I had just recorded these samples from the previous day. And what I'm doing is now I've finished this plot. I have finished all of this transect as well. So I'm going to group it together. And then I'm going to roll them so that they stay together. And from there, we're gonna put it inside of our paper bag. And then since this paper bag is now done, I've processed all the samples from this transect. I'm going to put it on this shelf. Yes, I am short, and yes, the shelf is a little high for me, but make it work. And I just worked on this basically all day. And this is a cute chipmunk I saw during lunch. I think we're gonna be friends now. He's so cute. So Thursday morning is measuring day. This is me walking into the nursery, and it's right outside of the greenhouse. So basically what you're gonna see is I'm going to use these carts right here. We see the tops of them. We are going to put our plants on this, and we're gonna wheel it to the shaded area because it is hot today. This is Edgar again. We're just measuring, we're looking at plant height and leaf number. And this is for our priority effects project, which is essentially we planted the first round of plants in early June, and then 3 weeks later we planted the rest. So we're looking at competition between plants to see if it matters, the order of arrival for establishment in restoration purposes. And look at this super cute slug we saw. So that took us all day on Thursday, and then I had to continue the next day on Friday by myself. I'm really tired because I went to a concert and my phone overheated because we had to do it in the full sun today. But that was my week as a RU student working restoration at the Chicago Botanic Garden.